Hi everyone, I'm very happy to be here online to share my short Löwdin lecture, the one for students. And the topic I decided to present is building database, and especially showing you that it's not always boring to build database as it might look. And the reason why I selected this topic is that it's very complementary to the longer Löwdin lecture that I will be giving on December 6th, and very complementary, but not at all addressed in the longer talk. You see here a list of the key people who contributed to this work, and you will see them again later, and I will come back to their contribution when in due time. So just few learning outcomes that I would like you to remember at the end of this talk. When I was a student, I actually had no clue about what did bottom up and top down meant, even if I actually heard a lot about this terminology. So I hope you will understand it a little bit better in case you're not familiar with it. I will show you that databases are super important. I will also mention that we cannot do everything with artificial intelligence, and that human intelligence was still relevant in this work. And I will also mention a couple of tools. Maybe worth mentioning that the effort of building database is also very present in some of the other speaker in the Löwdin symposium. So I invite you to follow also their work on the topic, especially David Balsell and other colleagues regarding the construction of transition metal complexes database. So here I start with the same figure as I will start in my other talk. This is basically an inverse design workflow, and many different components required attention to have a functional workflow. Today, I won't discuss all these components. I will discuss especially the one that you have here, that's on fragment database, but always the goal in inverse design workflow is that you really want to target a property, and you try to find what's the optimal molecule that give you, that possess, if you want, this property. We, especially ourselves, use a genetic algorithm for the optimization in the inverse design process, but not only, but it's one of the algorithms that we use. And this algorithm, you really need to have fragments that are called genes that you can reshuffle together to have your final molecule. And then you reshuffle this fragment, always evaluating the fitness function. That's something that I will not discuss, but that tells you if your new molecule is better or worse than the preceding one. You then proceed with crossover and mutation until your fitness function gives you a very good satisfactory value. But the key point that is related to the present talk is that you have to define the fragment, and you have to define the genes, if you want, how this fragment or this gene can be assembled together. For that, there is two main strategies in general. One is the bottom-up strategy. In the bottom-up strategy, you have actually building block that you can, that you know and that you decide that are good. And then you need to specific rules to decide how to assemble this building block into your final molecule, that all you assembly of building block constitutes the candidate space that you will then navigate to identify which molecule that is made of building block is the best in terms of property. The limitation here is that it's biased, because you have to predefine this fragment, which might prevent novelty, because also the way you assemble this fragment can be biased. And you need these specific rules to assemble this fragment together. On the other side of the spectrum, you have this top-down strategy, where here you start actually from existing repository, for instance, the Cambridge structure database that contain molecules that have been synthesized. And you find a way or define a way how to disassemble this kind of molecule to have building block that you can then reassemble. In this case, the limitation are that you lose a little bit of the chemical intuition, because when you have fragments that you can systematically modify, either a substituent or a scaffold, you will really know which component affects correlate the best with a given property And the chemical intuition will be then used to even improve the molecule that you're trying to build with a specific property. When you have the full molecule that is not originally based on fragment, you lose a little bit this intuition. Those databases are very heterogeneous. They contain whatever experimentalists will put in. Experimentalists or also theorists in some other example. So they are not always clean and strategically built, if you want. And heterogeneous database could also be a problem for inverse design or machine learning effort. They could be error prone because, again, experimental structure can contain error. So that's something you cannot master. So what we do in the effort I will show you, it's a combination of bottom-up and top-down approach. And what we're going to tackle is a specific field that hasn't been well covered so far with database. The way we generally build database really reflects the way we think about structure. It's, for instance, very easy to have automated workflow to construct transition metal catalyst. Here is an example from Jan Janssen from 2020, where transition metal catalyst, you have to think of them in terms of a metal to which are attached some ligands, and those ligands might contain substituents. So then you can enumerate a set of ligand, a set of substituent, a set eventually of linker and transition metal, and reshuffling them. And by doing so, you can have quite easy automated workflow to construct this transition metal catalyst. It's a little bit similar with biocatalyst. They have a lot of modularity, which is what we are looking for when building structure from the bottom up. So you can have an ensemble, of amino acid, and you shuffle them, and that will help you to build a biocatalyst. Organocatalysts, however, which is the topic I'm going to focus on, they are not thought as being modular. So the way people think of organocatalysts is to have few privileged catalysts without really modularity or fragment component that could be reassembled. And they are also thought in terms of substrate activation mode, meaning how the catalytic activity, where is the catalytic activity coming from the structural point of view, but no modularity. So you can classify them according to catalytic motif, Lewis base, Lewis acid, covalent pattern, non-covalent pattern, but an exhaustive enumeration is very difficult. And for this reason, the fragment-based approach hasn't been harnessed for those class of catalysts. And there was a lack of general strategy and platform for fragmenting, assembling, and navigating across the really wide chemistry of organocatalyst space. And that's what OSCAR is about. OSCAR is a database of organic structure for catalysis repository. It was thought and designed by my former PhD student, Simone Gallarati. As you will see, it covers functionally and chemically diverse catalysts. It's fragment-based, and it can be enriched in a combinatorial manner. So how does that's the most important slide, I will say, that really explain the workflow that we have been using, hybrid, top-down, and bottom-up. And it starts with using the brain of Simone to actually build this, not to extract 1,000 catalysts from literature and from catalog like Sigma Aldrich as a ChemDraw, and he extracted 1,000. And the reason why he did it manually, because he did this so-called seed database manually, is that there's about 1,500 publications on organocatalysts per year. And it's quite difficult, if not impossible, to really curate all the existing paper. But Simone, what he wanted to have is to ensure that the database, the seed database, cover enough chemical diversity in terms of chemical functionality, catalytic motif, scaffold, substituent, but have something that makes sense synthetically and that is accessible. So he got this 1,000 compound converted into SMILE and XYZ. And then that's the most important step. He listed function-based fragments based on this seed database. And the key challenge here, you have all the SMILE of these 64. 64 is totally arbitrary, but that's what he enumerates. And his challenges here were really to define pattern that it could then search in the Cambridge structural database to really enhance this seed database. And he needed this fragment that could return enough organocatalyst-like entries, but not too many general organic molecule entries. And this was, again, an effort that is based on the human intelligence and not on the artificial intelligence. We were not able really to find a good way to do it using artificial intelligence, and Simone did it actually better. Once he had the 64 fragments, he could actually search these catalytically active fragments in structure that comes from the existing CSD repository, so the Cambridge structural database, regardless of the structure were known as organocatalyst or not. The assumption was that if we extract structure from the CSD that contain these fragments, they could potentially be organocatalysts. And this leads to 3,000 additional structures that were extracted as 3D, XYZ in the Cambridge, and converted into SMILE. That's the top-down effort that we had. And to make this step easy from the 64 fragment and search in the crystal database, we used a software in the lab that is called Cell2Mol that is able to retrieve 3D info from CIF file in the crystal database. Here is a slightly more detailed description of how Cell2Mol work. It can retrieve Cartesian coordinate connectivity, SMILES, total molecular charge, metal oxidation state and spin, and get all this information to build several databases that you see here and that I will not enumerate. So bottom line is that when we are in the top-down end of this process, we have a database that is not exhaustive, but chemically diverse with commercially available and synthetically accessible compound augmented with what exists in the CSD. Here is just an example of how the enrichment affects the seed database. You see that some functional group, like this dual hydrogen bond pattern and this amino functional group, get enriched going from the seed to the CSD. That's not very surprising. The CSD contain a lot of pharmacophore that contain phosphorus and amine group, and this causes this kind of enrichment. However, in terms of size, you see that we don't have a huge increase in size, in molecular weight of the compound that are contained in the original seed database and those that end up in the enriched database. Here is a little bit of a structural map of what we have, and as you see, and it's very common, very typical of organocatalysts, there is no correlation between the 3D structure. So this is let me tell you, sorry, in a little bit more precise manner. It's a TSNE plot, so it's a dimensionality reduction using as an input the 3D structure, if you want, or a post-process information based on the 3D structure that is then mapped and colored by the different functional group. And as you see, all the color are pretty much present everywhere. The x-axis more or less indicate the degree of conjugation of the compound that we have, and the y-axis more or less the size of this catalyst, and there's no real correlation between 3D structure and motif. This is something that we would not see in transition metal catalysts that have actually more of this correlation. In addition to providing the structure of this so far 4,000 catalysts, we also have some kind of property. Here you have electrophilicity and nucleophilicity that can be computed based on conceptual DFT. And I won't go too much into detail of the property map, but I think that's important to note that those databases are somehow labeled, meaning that we have both the structure present and some property that are interesting to characterize organocatalysts in general. And I think nucleophilicity, electrophilicity, buried volume are those kind of properties that are available. We have everything available on Chemiscope. That's an example, a snapshot of this picture, but in principle, you can see the 3D structure. You can see a TSNE map and also which property have been computed for a given catalyst, property that correlate with the catalytic activity in some example that I won't have time to discuss today. Let me briefly, because I know I should give a short talk, but talk about, okay, we have this 4,000, so 3,000 for 1,000 structure. That was the top down. How can we enrich it from the bottom up? Where we enrich it by fragmenting this database using different philosophy depending on the type of catalyst that we want to focus on. I will show you an example. In one, we have been able to fragment these 4,000 catalysts based on some criteria. We can reshuffle them in a combinatorial manner and have this Oscar-enriched bottom-up subset or superset, if you want, of those organocatalysts. And I will give you briefly two examples. And for all of this, we always have the 3D structure and some kind of computed electronic property at different level. So just to show you one example, here it was on the carbene. The way to proceed with this bottom-up is to extract all the carbene that we had in the seed plus CSD database that corresponded to 195. They were extracted, they were analyzed, and they were decomposed into 17 central ring systems that were identified, plus different substituents and different substituent pattern. And based on this extraction and analysis that were done partially manually, partially automatically, the SMILE were generated, and if you want fragment SMILE, so that all the different components present in these three boxes here, scaffold, substituent pattern, and substituent themselves, were reshuffled in this Oscar NHC, so carbene on the nitrogen-based structure that contain about 8,000 carbene. We have here some kind of property that are relevant when you analyze the activity of carbene. You want carbene to be quite nucleophile and react quickly or strongly with an electrophile, but you also want them to have some kind of protected environment, asymmetric to leads to asymmetric catalysis. And that's kind of what this plot is showing here, even though I don't have much time to go into the chemistry. But here, the two properties that are key are the nucleophilicity and the buried volume that are typical as seen in this publication here from the Nolan group. Another strategy was for the dual hydrogen bond donor. Here we extracted from the seed plus CSD database about 1,500 structure. We identified this time using graph, molecular graph, what were the motif of this dual hydrogen bond catalytically active fragments. Once we have this motif, we identify 7 catalytic motifs and about 600, close to 700 substituent on the right and left-hand side. We were able to also recombine them. This leads to a combinatorial explosion because we had many more substituent on the right and the left side. So some of those, about only a subset of 6,000, were computed with DFT. Again, one descriptor being sterically relevant, one descriptor being electronically relevant. And in organocatalysis, you always need a good balance of steric and electronic factor. And those are the things you see on this map here that I don't want to discuss. But all of the structure were optimized, at least at the DFTB level, which is a lower level electronic structure level, given the number, the million of catalysts that we have been generating. But for a big portion, we had also DFT optimized structure. Very quickly, we have another existing database that is called FORM, that is based on the same philosophy of going top down from this Cambridge structural database, and then reshuffling and having recombination of fragments that are derived from the top-down process to reshuffle in a combinatorial manner in the bottom-up. That's a database that was designed for organic functional material like singlet fission or singlet triplet inversion. And this effort was made by Terry Blaskowitz, my other PhD student, and I won't discuss that today. Here's just a few examples of application, actually, of this catalyst on proper inverse design problem, in case you are interested. The first block is for organocatalysis, the second block is for organic electronics. And I hope you learn a few things here. You learn what does bottom-up, top-down strategy and hybrid strategy means to build those databases. I show you that it usually combines human intelligence and automated pipelines. Those are repository of structure, but also of property. And they are also simply a library of fragments that can be assembled in the inverse design genetic algorithm pipeline. What we're trying to analyze now is that, is there a good alternative to humans' decision? Because we thought the humans' decisions that were made were good. And how to avoid modularity, for instance, using generative models instead of genetic algorithms. So stay tuned if you're interested in that. That's what will come out of the group soon. And with that, I want to thank the whole group. This is a semi-updated last slide with the people involved, especially in this effort, Simone Gallarati and Terry Blaskowitz for the two databases, Ruben Laplaza and Sergi Vela and Yuri Shaw that were involved in the tools that I mentioned. And I would like to thank you for your attention, and sorry for being slightly over time, although not too much. I could have talked much longer on the topic, but I hope you got a kind of a understanding of how this effort could actually be more fun than it sometimes look, and also absolutely essential in the current era of big data and data-driven workflow for molecular design. And I thanks all the members of the group and you for your attention.